Hello, everyone. My name is Louise, and I will be giving today's Ensembl browser workshop. So, first of all, I've just wanted to introduce you to the European Molecular Biology Laboratory, or EMBL. Oh, apologies. There are 6 sites in Europe. There is Ensembl EBI, which is where I'm based at. It's one of our biggest institutes. We've got a site in Grenoble in France, which focuses on structural biology. Another site in Barcelona in Spain, which focuses on tissue biology and disease modeling. We have 2 in Germany, one again structural biology, and the other our main site is in Heidelberg. This one focuses on life sciences in general. And then our 6th site is in Rome in Italy, which focuses on epigenetics and neurobiology. The European Bioinformatics Institute, the EBI, this is where I'm based at. So we are based in the UK, and we provide free bioinformatics resources for scientists around the world. We also have a few research groups in computational biology, and one of our main tasks is to provide bioinformatics training. You've probably seen some of these resources. The Ensembl EBI takes care of the European Nucleotide Archive, for example, ENA. This is a database where you can upload your own sequences. We've got Open Targets, UniProt, and one of our biggest resources, I'd say, definitely is Ensembl, which is a genome browser. At the end of the workshop, I hope that you'll be able to describe what Ensembl is, how to navigate the online browser, how to access the different data that you can find in Ensembl, and most importantly, where to find documentation and where to go for help. You can find all training materials at training.ensembl.org. You can also follow the link at the bottom of my slide here. And these training materials will be available to you indefinitely. So that means that even in, let's say, 6 months' time, if you want, you can still go back to the exercises and try them out again. On the training page, you'll find the slides, a link to the living document. So this document just lists down a few resources that might be helpful to you. And we've also got the exercises with answers in them. So we're going to go through an introduction to Ensembl. I'll also talk about how we annotate genes and transcripts. Next, we will talk about variation data and the variant effect predictor, and then we'll briefly look at regulation data in Ensembl. So just a bit of background and an introduction to Ensembl. So in 1977, Fred Sanger and his team sequenced the very first genome, which was a bacteriophage. And this really kickstarted the era of sequencing and sequencing much more complex organisms. The first draft of the human genome was published in 2004, which was a project that was started back in the 1990s. And since then, many more organisms have been sequenced. And today we can sequence a complete human genome in a matter of days. With this, Ensembl was launched in 1999 with the goal to automatically annotate this very first draft of the human genome and also integrate it with other data and make it publicly available. But why do we need genome browsers like Ensembl? Sequencing has become much cheaper and faster, and because of that, genomic data is quickly piling up. But when sequencing data is produced, you get something like this. So just a bunch of A's, T's, C's, and G's. So trying to read a genome without annotation data would be like trying to read a new language without a dictionary. It wouldn't mean anything to you. And this is what genome browsers are there for. Genome browsers map annotations and different features to genomic regions, giving the genomic region a value. And what Ensembl does is it brings together information from a wide range of other databases into a single site. Other genome browsers you might have heard of are NCBI and UCSC. And I want to emphasize that Ensembl does not do its own sequencing. So we simply take publicly available sequencing data and we use our own pipelines to annotate the genomic data. There are many features that are annotated by Ensembl. So these include genes, variants, promoters, binding sites, and many, many more. We have variation data in a number of different species, comparative genomics data, so alignments, gene trees, and homologs. There are also regulatory builds available for human and mouse from experimental data. You can use a number of different tools in Ensembl to, for example, export large amounts of data like Biomart, or process your own data to find out the effect of your consequences using the variant effect predictor or VEP. You can also access the Ensembl database via our APIs. And Ensembl data is completely open source. That means that you can access all of the data that we display in Ensembl completely free. And you can also access all of our source code in our GitHub repo. As genomic data is constantly evolving and changing, Ensembl is updated every 3 months to represent the most recent findings. Behind every update, a lot of work is involved, which includes the addition of new genome assemblies, updating underlying software, updating gene sets, adding new comparative genomics analyses on genes and genomes, updating variation data and regulation data, and adding any new interfaces. The current release is Ensembl 110, which was published back in July 23. And the next release, 111, is expected to be released or published in November 23. And because Ensembl is not static, you want to make sure that you write down what Ensembl release you are working with. We keep archives for up to 5 years online. After 5 years, we take the website down and the data will only be available on our FTP site. To keep up with the constant release of new genome assemblies, we also have an Ensembl rapid release browser. So this is updated every 2 weeks. And this browser has limited information compared to the Ensembl's main site. It includes genomes with gene annotations only, and the only tool that you can use is BLAST. And some comparative genomics data is gradually being added. We also have the T2T genome in the Ensembl rapid release site if you're interested. So we know that genome assemblies are not a perfect representation of the genome, and there are still gaps in genome assemblies, but researchers fill these gaps, correct errors, and share patches or updates continuously. And this means that there can be different versions of genome assemblies. Ensembl has 3 versions of the human genome. We've got the GRCH38 or HG38 genome. This is the most recent and also the most supported human genome assembly. And it has the fewest gaps, and many rare alleles have been replaced with more common ones. We also have a dedicated portal for the older GRCH37 or HG19 genome assembly, as this is still widely used in research. So this assembly has some larger gaps, and there are only limited updates and data available for this. Finally, we also have a dedicated site for the NCBI 36 or HG18 genome assembly, but this has even more gaps and it is no longer being updated. Okay, next I want to talk about how we annotate genes and transcripts in Ensembl. Um, so genomes that are annotated with genes are referred to as gene builds. And this is a term that you will come across quite often when you work with genome browsers. There are two independent annotation methods that we use in Ensembl, but both rely on valid biological experimental data. So that means that we only add gene annotation when there is experimental evidence that the gene was transcribed. We have the Ensembl automatic annotation pipeline, which is applied to all vertebrate species. And we have the Havana manual annotation pipeline, which is done by a group of annotators. And this is only applied to human, mouse, and partially zebrafish and rat. The automatic annotation method is a gene prediction pipeline. So we take known data, for example, transcripts, proteins, cDNA, and RNA-seq data, and map these to the genome. It can take up to 2 weeks to annotate a single genome this way. The manual annotation method is done by an annotator, so a person, and it can take several years for a single genome to be annotated. As you might guess, the manual annotation process is much more laborious as the annotator moves along the genome to map published experimental data, and they use different databases as well. It can take up to half a day to annotate a single gene. However, manual annotation is much more accurate for difficult features. So for example, splice sites, but it takes much more time as well. This is why we only perform manual annotation on human, mouse, and partially zebrafish and rat. So this is a snippet of the region in detail view, which you will explore during the exercises, sorry. But what I mainly want to show you here is how you would see a gene on the Ensembl genome browser. So let's take, for example, the MAGT1 gene. You can see here that there are actually 5 annotations for MAGT1. So 1, 2, 3, 4, 5. And this is because in Ensembl, a gene is defined as a collection of transcripts. So many alternative transcripts can be transcribed from a single locus, and this is what you're seeing in this view. Each transcript can be expressed differently and can serve a different function. And there are different types of transcripts, and they are color-coded here in gray, red, blue, and gold. We have non-coding transcripts which do not produce proteins, for example, RNA genes. These are colored in gray. We have protein coding transcripts, which are transcripts that are translated and produce proteins. These are colored in red. We also have non-coding transcripts of coding genes. So these are transcripts of coding genes, but they are not translated. So for example, retained intron transcripts. These can be fragmented, or they may be missing a translation start site. They may be faulty, degraded, and so on. These are colored in blue. And then finally, we have the golden or the merged transcript. So these transcripts represent transcripts, this flag or tag represents transcripts with a high confidence in the annotation. These golden transcripts can be both coding and non-coding, and I'll talk more about golden transcripts in the next slide as well. Let's look at the individual features of a transcript. So let's focus on this very first transcript of the MAGT1 gene here, this golden transcript. So here you can see that we have these lines and these boxes. So the boxes represent exons. Empty boxes are non-coding exons. And the filled boxes, like this one here, for example, is a coding exon. And the lines represent intronic regions. If you look closely, you will notice that this line is kind of V-shaped or valley-shaped, and that just tells you the direction of the transcript. So on which strand the transcript is located on. So if it's a V shape or a valley shape, it means the transcript is located on the reverse strand. If it's A-shaped or hill-shaped, like this transcript up here, this means that the transcript is located on the forward strand. You can also see on which strand the transcript is located on by looking at the label or the name of the transcript. So if we focus on this very first transcript here, this has a greater than sign. This means that the transcript is located on the forward strand. And if we go back to our golden MAGT1 transcript here, this one has a less than sign. That just means the transcript is located on the reverse strand. There are several levels of confidence in the annotation of genes and transcripts, and we always flag the annotation confidence in Ensembl. Golden transcripts are transcripts that have been annotated identically by both the Ensembl automatic and the Havana manual annotation methods. This means that the features of the transcript where the annotation was done, it was annotated identically by two independent annotation methods, meaning that there is a higher confidence in the function and the quality of the transcript. We also have what we call the GenCode gene set. The GenCode project focuses on the Ensembl human and mouse gene sets, and it is the merged gene set of the Ensembl automatically annotated and the Havana manually annotated genes in human and mouse. It's also the default gene set used by many major projects like Nomad, Decipher, 1000 Genomes Project, and so on. Another project to know about, and which also dictates annotation confidence, is the main select project. So main stands for matched annotation from the NCBI and Ensembl EBI. And it is another measure of the quality of a transcript. So NCBI has its own gene annotation pipeline and a gene set called RefSeq. And main select transcripts are just transcripts that are 100% identical in both RefSeq and Ensembl. The main select transcript is agreed to be the most biologically relevant and the representative transcript for a gene. Elaborating on transcripts, I also want to introduce you to canonical transcripts, which are a single representative transcript identified at every locus in every species. So these are transcripts that are most highly conserved and most highly expressed. They are in concordant with other transcript scoring algorithms and UniProt canonical isoforms. They have the longest coding sequence, the largest number of clinically important variants, and they are complete. And wherever available, the Ensembl canonical transcript is usually also the main select transcript. All features in Ensembl have Ensembl stable IDs assigned to them. This is composed of an ENS prefix, a single letter representing the feature type. So for example, G for gene, T for transcript, and so on, and an 11-digit unique number. Stable IDs do not change between releases. If there is a minor change, we would increase the version number after the dot. For any non-human species, we also add a 3-letter code between ENS and the feature type. So for example, for mouse, we would add MUS, and for zebrafish, we would add DAR. So if you see any IDs that look like the ones up here, this means that these are all human features. When we annotate genes in Ensembl, we also always provide you with any linking information about them. So these include Go terms, gene ontology terms, so the function, any protein domains if it's a protein coding gene, any links to other databases, a list of variants within the genes, a list of phenotypes associated with the gene, and any homologs of the gene in other species. Okay, next I want to talk about the variation data in Ensembl, and we're going to look at how to use the variant effect predictor, or VEP. So let's talk about variation types first. We can group variants according to their size or the number of nucleotides that are affected. Variations can come in a small scale where only one or a few nucleotides of a gene are involved. So here on my slide, we have two sequences. This top sequence, let's call this our reference sequence, and our bottom sequence is our own data. So small scale variation types include small insertions or deletions. We also call these dips or indels. And we also have single nucleotide polymorphisms, or SNPs, which are changes in the nucleotide. So in this example, the reference sequence has the A variant or A allele at this position, and in our data, we have the T allele. Next, we have an example of a deletion. So in our reference sequence, we have a C allele, and in our own data, we have a deletion at this position. And the third example is an example of an insertion. So in our reference sequence, there is no base related in this position. And in our own sequence, we have an insertion of a G allele. Variations can also come in large scale, for example, in the chromosomal structure. So these are also called structural variations, and the types of structural variation are copy number variants, or CNVs. So this is when you have a genomic sequence with a lot of repetitive regions. We have large deletions, duplications, insertions, and translocations. So now that I've given you a brief introduction to transcription, our variation data, I want to talk about how Ensembl processes the variation data. So first, we import the data. This will always come from public databases because we want to make sure that all of our users have the exact same access to the data. Next, we perform some quality control measures. Then we link the data to other databases. So, for example, we link the imported variants to allele frequencies, to any publications, to any phenotypic data, and so on. And then we perform our own analyses on these variation data. Some of the data that we link with the variation data are phenotype and disease data, and these can come from a range of different sources depending on the species. So for example, for human, we link variation data to the Human Gene Mutation Database, or HGMD, and also ClinVar. For mouse, we link variation data with phenotype information from the International Mouse Phenotyping Consortium, or IMPC. and also the Mouse Genome Informatics Database, MGI. And you can find the different sources for each species in the link at the bottom of my slide. We also collect information such as the clinical significance of the variant, inheritance type, the reported gene and risk allele, and for genome-wide association studies, or GWAS, we also collect statistical information such as the p-value, odds ratio, and beta coefficient. So linkage disequilibrium is a measure of how much the presence of one allele influences the presence of another at a different locus. Certain combinations of alleles may occur more or less frequently in a population than would be expected from a random formation of haplotypes. And this non-random association can be influenced by many factors, including selection, the rate of genetic recombination, mutation rate, genetic drift, population structure, and genetic linkage. Linkage disequilibria, or LDs, are often visualized in what we call an LD plot. And these plots will show you a measure of how correlated specific loci are. So you would often get these measurements in R squared, and an R squared value of 0 would indicate that the loci are in complete linkage equilibrium, so they are not co-inherited. And an R squared value of 1 would indicate that the loci are in complete linkage disequilibrium, meaning that they are co-inherited. So I mentioned in the variation data process, in the Ensembl variation data process, that in the last step, we apply our own analyses to the variance. And what we do in this step is we predict the consequence of the variance that we've imported. So what we do is we take each variant and we plot them along the genome. And we predict the functional consequence of the variant depending on where the variant falls within a gene. So on my slide, you're seeing a gene. The lines represent intronic regions, the boxes represent exonic regions, and these yellow boxes, or these filled boxes, represent coding exons. So if a variant falls outside the gene, let's say the 5' or the 3' region, it's unlikely to affect the gene itself, but it may affect regulation. So for example, mRNA transcription or transcription activity. If the variant falls within a gene, but not within a coding sequence, so let's say any intronic regions, it is also unlikely to affect the gene itself, but it may affect mRNA stability. So for example, splicing or translation efficiency. If the variant falls within the coding region of the gene, so any yellow boxes on here, the variant can have two effects. It can have a synonymous or a non-synonymous effect. And if the variant has a synonymous effect, the protein sequence remains the same. So there's no change in the amino acid. If the variant has a non-synonymous effect, there is a change in the amino acid and therefore a change in the protein sequence. So this means that the variant is likely to have a much greater consequence to the gene and the protein function itself. We also call non-synonymous variants missense variants. So to summarize, in Ensembl, we plot each variant to the genome, and depending on where the variant falls within a gene, we assign a consequence to the variant. I don't expect you to read this table, but what I want to show you here is that variant consequences have special terms, which we call sequence ontology or SO terms. and it is just a standardized way of describing the consequence of a variant. Just like how we have a standardized way of describing a gene function with gene ontology or Go terms. When we talk about non-synonymous or missense variants, we rank them by their pathogenicity. There are different algorithms that rank these changes in the amino acid sequence based on how conserved the protein is, the chemical change in the amino acid position, the 3D structure of the protein and its domains. So all of these can dictate the protein function. However, please note that these are all predictions and not facts. So it's always good practice to validate your predictions experimentally. So the human reference genome is not a genome made of a single person, but it is actually made up of fragments from different individuals. So when we take a sequence from the reference genome, let's say we're taking this sequence from this reference genome, this sequence is actually found from this individual. So when we look at a specific variant, so for example, this T in this sequence, it doesn't necessarily mean that it is a representation of all the human individuals around the world. This T allele is found in this particular individual, but it does not necessarily mean that the majority of individuals actually have this T in this position. This individual who's carrying the T allele might, for example, actually be susceptible to a particular disease. So when we talk about reference alleles, they aren't always the major allele or the allele found most frequently in a population. But most of the time, the reference allele is the major allele. It's just something to keep in mind. And if you have a list of your own variants, for example, let's say you've done your sequencing, you've called your variants from your sequences, and now you actually want to find out the consequences of your variants. You can use one of our tools called the Variant Effect Predictor, or VEP. So what you can do with VEP is you can find out the effect of your SNPs, indels, and also structural variants. And this is how you use VEP. So first, you need to enter your data into one of these formats. Your data is then processed by VEP and searches Ensembl and external databases to give you information such as the effect of the variant, the pathogenicity score, if it's a missense variant, any allele frequency data, consequences, whether the variant falls within a regulatory region, and any associated publications and phenotypes as well. There are 3 different interfaces for VEP. You can use the web interface on the Ensembl browser. You can access VEP through the command line, or you can access VEP via our REST API. When you retrieve your VEP results, you might get overwhelmed with the amount of data that it gives you. You can filter your results to prioritize the variants that you are actually interested in. So here are just some things you might want to focus on when prioritizing variants. You may want to limit your variants to the ones that are located on certain genes, or more specifically on certain transcripts. Maybe you decide to consider variants that are located only on transcripts that have a certain transcript support level, which is just a scoring system of transcript annotation confidence. You can limit your variants that are found on GenCode gene sets. So this is a set of evidence-based gene annotations in human and mouse. And it is the merged gene set of the Ensembl automatically annotated and the Havana manually annotated genes. So these two independent annotation methods we use in Ensembl. And it's also the default gene set used by many major projects like NOMAD, 100,000 Genomes Project, or 1,000 Genomes Project. You can also consider variants within main transcripts. So the main project is a collaboration between the NCBI and Ensembl EBI, and these are transcripts that are labeled these transcripts that are labeled as main select transcripts just flag high confidence within the annotation. So they are a set of representative transcripts for each gene that were identified independently and, and annotated identically by Ensembl EBI and NCBI. Main select transcripts are also determined to be the most biologically relevant, and main plus clinical transcripts specifically include any additional transcripts that are found to be of clinical interest. So, for example, transcripts that are pathogenic or likely pathogenic. Maybe you're interested in variants that are associated with certain phenotypes or traits only, or maybe you want to focus on missense variants that are predicted to have greater effect on the protein. Or maybe you are looking for rare variants or even want to find out if any of your variants are novel or have been annotated before. So to help you with prioritizing your variants, VEP supplies you with a range of information which you can use to filter your results. VEP will report any genes, transcripts, and regulatory elements that your variants map to. VEP will tell you if any of your variants have been reported in databases like dbSNP or COSMIC. We also report any allele frequencies from the 1000 Genomes Project, exome sequencing project, NOMAD. So if you're interested in rare variants, you can apply stringent allele frequencies to filter out any common variants. For missense variants, you can retrieve a variety of pathogenicity scores, and we report any associated phenotypes, traits, clinical significance, and publications if available. So next, I want to talk about regulation data in Ensembl. In Ensembl, we have a number of genomes that we've annotated with regulatory elements or features that control gene expression. So, for example, promoters, enhancers, and repressors. And to annotate these regulatory elements, we look at specific data types like epigenetic marks, so histone modifications and DNA methylation. We look at transcription factor and RNA polymerase binding sites, or we look at open and closed chromatin conformations by looking at DNase 1 sensitivity. So let's look at how genes are regulated. Essentially, each cell of an individual shares the same genome and therefore the same genes. But we know that there are hundreds of different cell types with very different or very distinct phenotypes. And this is because of different expression levels of different genes. So if we consider the diverse number of blood cell types, for example, we have, they all have very distinct functions. We have red blood cells, which are important in oxygen transport. We have basophils, type of white blood cell, which are involved in allergic reactions. We have B lymphocytes, which are involved in antibody production. So all of these cell types originate from a single hematopoietic stem cell. And what makes blood cell types different is how they express genes at different levels and at different times. We say that these different cells have different profiles of epigenetic expression or epigenomes. You can think of these epigenetic modifications as layers of transcriptional regulation or layers of controlling the different timings and expression of genes, which is what you're seeing in this diagram. So on the slide, you can see a strand of DNA. At the bottom left, we have the coding region of a gene. And upstream of the gene, you can often find the promoter region, which binds RNA polymerase, so the enzyme that transcribes DNA into mRNA. The promoter region also binds transcription factor proteins, so these are regions where these regions we also call transcription factor binding motifs or transcription factor binding sites. And these transcription factors are involved in inhibiting and stimulating the transcription of a gene. Another regulatory element are enhancers. So these sites are often located a bit further away from the coding region of a gene. And enhancers have their own set of transcription factor motifs, and they bind activator proteins which promote the activation of transcription in the promoter region. Similarly, we also have repressors and insulators that bind specific proteins to downregulate gene expression. The binding of these transcription factors is regulated by methylation. So within the DNA sequence, we have regions that are called CPG islands. And these regions are rich in CG repeats. And what happens is DNA methyltransferase moves along the genome and modifies the C bases within these CPG islands with methyl groups, so CH3 molecules. And this gives different methylation patterns, and these different methylation patterns can modulate the binding of transcription factors. So for example, methylation can block a transcription factor from binding to a transcription factor binding motif. So DNA methylation is just one layer of transcriptional regulation, but there are many other layers as well. For example, the conformation of the chromatin can direct the activation or deactivation of a gene expression. In this diagram, you can see that the chromatin is bound in a uniform manner. And the DNA strand is twisted around 4 histone proteins. So that's what you're seeing here, making structures that are called nucleosomes. And regions of densely packed chromatin are associated with transcriptional deactivation because the DNA is less accessible to transcription enzymes. Regions of chromatin that are more loosely packed are associated with transcriptional activation because the DNA is more accessible to transcription proteins. We also know that the position of the nucleosome and the modification of histone tails, so post-translational modifications like acetylation and ubiquitination themselves, can affect the transcriptional regulation. And then finally, the actual 3D conformation can also regulate gene expressions. So these mechanisms together with transcription factors and chromatin remodeling factors exhibit very unique epigenetic states which result in alterations of gene expression that can, for example, determine cellular diversity within DNA with essentially no difference in the DNA sequence. So these layers of transcriptional regulation can be assessed by different experiments which look at the expression patterns of the gene. And this diagram shows you the different techniques that can be used to determine these layers of transcriptional regulation. For example, we can use DNA sequencing to assess open or closed regions of chromatin by looking at the sensitivity to the DNase enzyme. We can use ChIP-seq to understand protein binding patterns and histone modifications. And we can also determine gene expression levels by looking at RNA-seq data. Now that I've given you an introduction or a brief introduction to transcriptional regulation, I want to talk about the process of how Ensembl uses this kind of data to annotate regulatory elements in the genome. So first, we import the different types of data from different sources. Again, the important part here is that the sources have to be open access so that all of our users have equal access to the data. Then we process the data to predict the positions of the regulatory features, so the promoters, enhancers, or transcription factor binding motifs. Next, we assess the activity of these different regulatory features in different cell types. And then finally, we display the regulation data on the browser. There are three main projects where Ensembl takes regulatory data from. One is the ENCODE project, which looked at 48 human and 8 mouse cell lines, and it generated data that looked at histone modifications, transcription factor binding sites, RNA polymerase activity, and DNA sensitivity. Another source is the Roadmap Epigenomics project, which looked at gene expression in cultured primary cells, and the Blueprint Epigenome project, which generated ChIP-seq and DNA sensitivity data of 20 primary hematopoietic cells. I want to highlight that Ensembl does not link regulatory features to genes, so we only provide you with the locations of the features. We also do not link regulatory features to gene expression, but rather we present cell line specific regulation data and tissue specific expression data. So when you are using Ensembl, we do not make inferences about the regulatory data, and it is up to you to make your own inferences.